Now we're going to hear from Katie Heyob. Presentation, the biodegradability of organic additives in hydraulic fracturing fluids. 2013, Katie received a BS in Geology and Environmental Studies from Eastern Kentucky University. She is currently a second-year MS student at The Ohio State University in the Department of Civil, Environmental, and Geodetic Engineering. Her research involves tracking the biodegradation of organic additives used in hydraulic fracturing fluids under anaerobic aquifer soil conditions, with a particular focus on the degradation of glycol formulations in these fluids, as well as the microorganisms responsible for these transformations. So Katie, thanks for joining us, and we'll turn the mic over to you. I guess that's the... That's your clicker, okay.
Is there a pointer on?
I think.
Okay. Well, okay. Right. I am very excited to be here today to talk about not only my master's research, but also the research of my fellow graduate students at Ohio State who have already graduated and moved on. But before I dive into my talk, I'd like to acknowledge them. So Shuai, Mariam, and Dan, they were master's students. Shuai now works at Halliburton in China. Mariam is at the EPA in Washington, D.C., and Dan works for a consulting company in Columbus. Maggie was an undergraduate student who's now studying at the University of Colorado Boulder. And I would also like to acknowledge my advisor, Paula Mauser. The funding for this work is provided by the National Science Foundation and the Ohio Water Development Authority. So one of our main research interests in our lab is to figure out the influence that microorganisms have in processes like hydraulic fracturing. So in order to understand this, we first have to recognize that microbes live everywhere, even in very, very extreme environments. So deep hydrothermal vents in the ocean and the Arctic permafrost, and of course, and shale formations in the subsurface. So what makes microorganisms difficult to study sometimes is that the majority of them are uncultivated in labs, so we have to alter our experiments to account for this. So more specifically, in shale energy systems, microbes of course, can produce methane and oxidize hydrocarbons, but oftentimes microbial growth is associated with fouling of our materials that we use for drilling. Gas wells are very, very good environments, I guess, for microbial growth. It's really warm, there's lots of organic carbon, and so when we are trying to optimize our chemical additives and fracking fluids, we need to try to limit the microbial growth, I guess. So, of course, hydraulic fracturing is this process of injecting fluids under high pressures to create fractures and aid in the recovery of oil and natural gas. And these fluids are primarily water and proppant with about 1% chemical additives. And if we zoom in on these chemical additives, we can see that they are providing a multitude of functions, such as inhibiting corrosion or reducing friction. And the majority of these additives are organic-based, and they range from common chemicals like alcohols, glycols, to surfactants, new polymers, things like that. So understanding how these chemicals are degrading in the environment naturally is very important. And it's important in case we have accidental spills. So there are a few main routes that I've highlighted here. The first is a surface spill, releasing these chemical additives to soils or the shallow subsurface. The second is leaks in protective casing, and the third, and maybe least likely, is migration upward from the target shale formation. And so for the purposes of our experiments, we like to try to simulate spills to the surface at this point, and then hopefully in future experiments we'll look into some of the deeper routes of accidental release. And so the EPA came out with a report in May of this year highlighting spills related to hydraulic fracturing, and most of these are flowback and produced waters, but some are related to the hydraulic fracturing fluids that are injected. Most of this is just container failure or equipment failure, so small leaks. And so since these releases may occur, it's really important to understand how these chemicals are degrading naturally and if they pose a risk at all. So in our lab, we like to think of this as kind of a scheme of higher overall bioattenuation potential and lower overall bioattenuation potential. And so if we think of conditions at the surface, say with a wastewater microbial seed, under aerobic conditions, we would expect the degradation of organic chemicals and fracking fluids to be pretty fast, compared to shallow subsurface or a groundwater aquifer where there might be lower microbial biomass and anaerobic conditions. And then from there, moving deeper into the ground, there's typically less microbial activity, and we would expect lower overall bioattenuation potential. So more specifically, we want to look into how solid media affects biodegradation, so soils versus like a sandstone, how microbial seeds influence biodegradation. So as I said, a wastewater activated sludge might degrade things faster than, say, a groundwater microbial community. How does high salt affect biodegradation? Redox conditions, so aerobic versus anaerobic. And then really the frac fluid is this complex mixture of organics. So there have been studies looking at the degradation of these chemicals as individuals, but when in a complex matrix, those rates and extents might be different. So in order to answer these questions, we typically don't have a lot of fracking fluid samples from the field. So we needed to develop a synthetic fracturing fluid that's representative of fracking fluids that might be used out there in the industry. So our first step was to develop this fluid. So on the left here, we have our synthetic fracturing fluid recipe. And the way we developed this fluid, we surveyed Ohio and Pennsylvania wells on frac focus, and we looked for chemicals that are commonly used in these disclosed recipes. And from there, we obtained the additives from chemical manufacturers. So once we developed our fluid and made it up, we then compared it to a sample that we did receive from the industry. And so what's interesting here is that we do have similarities, and we have differences as well. So in our experiments, we're looking at the degradation of organics. So we really want to make sure that our total dissolved carbon and our total organic carbon are statistically similar to the field sample. And in this case, we diluted our synthetic fluid, sort of like a low range, a middle range, and a high range, and compared it to the first column, which is the field supplied sample. And our low range fracking fluid was statistically similar as far as carbon content. But the differences we see are mostly in these elements. So this might be for a few different reasons. If this particular sample that we got from the field was made using flowback fluids, that might have an influence on things like chloride content, barium, calcium, things of that nature. And typically we see that those elemental concentrations are higher than our synthetic fluid. So with our experiments, we really target the carbon because those elements can vary so much between drilling location, operation, etc. So now that we have our fluid, we design a series of incubation experiments to look at the biodegradation of the organics in these fluids. And so the first study that I'm going to talk about is Dan's work. He looked at sort of a wastewater processing type treatment under aerobic conditions. And we wanted to address if wastewater activated sludge can even degrade the organic carbon in fracking fluids. A concentration effect, so more concentrated fracking fluid versus a lower concentration, varying salt concentrations, and then ultimately we want to see how much carbon disappears. Is the microbial community changing, and then is there a buildup of any specific compound as, say, polymers or surfactants are broken down. And so on the top here we have just an image of Dan's experimental setup. And below is the table just outlining his treatment. So we set these up in batch, and we introduce either low or high SFF. And as a positive control, we use low and high acetate, which is like candy to these microbes. They eat it up. And then we also have an abiotic control that's been sterilized to account for things like volatilization. And we measure DOC degradation. In this case, for his experiment, he did this for about a week. And so this first image is going to address these first two sort of variables influencing degradation. So in green, we have our synthetic fracturing fluid in high and low concentrations. The reddish color is the acetate, which is our positive control. And then our sterilized abiotic is in gray. And so, like we would expect, the positive control is degraded very fast, 30 to 50 hours. But what's even more interesting is that our low concentration of frac fluid is degraded almost the same as our positive control. In our abiotic sterilized controls, we see about 6 to 9% loss due to volatilization. And then in our concentrated synthetic fracking fluid, we have 43% dissolved organic carbon remaining after just about a week. So it's pretty promising that the dissolved organic carbon is degraded greater than 50% within a week in his experiments. So to address maybe some high salt conditions, Dan also added salt to a similar experiment. So he reran his treatments, added either 0, 20, 40, or 60 grams per liter NaCl. And he also used a lake water control just to see if maybe lake water microorganisms degrade things slower than activated sludge. So here we have dissolved organic carbon on the y-axis again, and time in days on the x-axis. He also measured microbial biomass through time, which is represented by the bar graphs on the bottom. And we see similar losses due to volatilization or absorption, 11% in our killed controls, which is in gray at the top. And then in our synthetic fracturing fluid with low salt, 0 or 20 grams per liter, we see that 43 to 49% is remaining after only about a week. And in high salt conditions, degradation is actually inhibited. There's no degradation of dissolved organic carbon. If we look into the cell growth through time, you can see that as those bar graphs really jump, it correlates with the dissolved organic carbon decreases, which is what we would expect. And cells, these bacteria are growing, which is promising. They're not getting killed off, and they're increasing about an order of magnitude. And if we do some yield coefficients and specific growth rate calculations, we can see that as 1 mole of carbon is assimilated into microbial biomass, about 4 moles of carbon are respired to CO2 in these systems. So what is happening with the microbial community? Is it changing significantly? And what we can do in our experiments is extract DNA from the entire microbial community, and we can sequence that and look at the diversity at, say, time 0 versus day 7. And so that's what we did. So for the sludge in the upper left and the lake water in the bottom left, those are initial time points where we sequence the DNA, and we can pull out the most dominant species in those systems. And what's interesting is in both the lake water treatments and the sludge treatments, the microbial community actually converges to about the same makeup of bacteria. And it's dominated by this Pseudomonas, which is really prevalent in like waste ponds and well fluids, which is pretty interesting that these both go to the same microbial diversity over time. We can also look at the buildup of specific organics in this system. So we took our samples, filtered them, and analyzed volatile organic compounds using an EPA method. And we can see that alcohols and limonene are detected the highest in initial concentrations. But what's really interesting is this huge buildup of acetone throughout the experiment that's really not going away. And when we calculate, so isopropanol typically degrades to acetone. And if we try to do a mass balance calculation, we see that there's much more buildup of acetone than isopropanol present, suggesting that the acetone is actually coming from a different parent compound. And this is interesting because we actually in the literature see that acetone is building up in other sites. So like in Colorado well flowback, there's high concentrations of acetone. Acetone can be slightly toxic, and it serves as a strong solvent in these fluids. So that's one research question that I hope to address in my research. So digging into this a little bit further. So to summarize Dan's findings, what we see is that yes, activated sludge microorganisms can degrade the organics in fracking fluid. Typically high concentrations have slower degradation, which is also what we would expect. High salts, again, slower overall DOC degradation. Much of the carbon is mineralized to CO2. The microbial community is dominated by Pseudomonas at the end of about a week. And we see this big buildup of acetone through time. So what about synthetic fracturing fluid degradation under maybe less ideal conditions, under conditions where oxygen isn't present? And that's really what I want to look into, and that's what I've been doing for the past couple of years, is looking at soil and groundwater microbial communities. And I really want to address the effect of matrix complexity, so this combination of organics, and also try to identify which compounds are degrading to acetone in these systems, and relating these two questions. So if I look at the MSDS sheets for the chemical additives that we use in fracking fluids, we see that the synthetic fluid contains various ethoxylated surfactants and polypropylene glycols, polyethylene glycols. And so polypropylene glycol specifically, if it's degraded, it would go to the monomer propylene glycol, and the microbial enzyme that's responsible for this is a dehydrogenase enzyme. From there, if the monomer is broken down, it'll go to its corresponding aldehydes, alcohols, and carboxylic acids. And what's interesting here, more specifically, it's a propionyaldehyde, but we see acetone could be produced from these glycol polymers. And then if everything's completely mineralized, we'll just see a production of CO2 or methane. And so I targeted two specific chemical additives that we have, a revert flow stimulation surfactant and an AI600 corrosion inhibitor for my experiments moving forward. And so my sort of overall hypotheses here is if we have a complex hydraulic fracturing fluid, it's going to degrade slower, it's going to maybe have more DOC left over at the end of the incubation period than individual chemicals like the revert flow corrosion inhibitor. If we move even more simply, propylene glycol monomer and the polypropylene glycol polymer, we would expect those to degrade faster. They're shorter organics, easier to attack from a microbial standpoint. So I set up a bunch of different treatments using soil and these substrates. And I used groundwater and soil microorganisms, of course. And all of my bottles are anaerobic, so there's no oxygen present in these. And so just to kind of visualize how my bottles are set up here, I have the solid media in the bottom, and then I add one of those treatments as 90 milliliters, and I set up hundreds of these things in triplicate so that I can sample them over about 50 days in 7 time points. And when I go to sample, I'll open them, sacrifice the bottle, and then use multiples. So all of these bottles are just kind of gradually stirring in the dark until I sample them. So for my results, I know there's a lot of lines here, but we have dissolved organic carbon on the y-axis again, time on the x-axis. For A, those are all biotic treatments, and for B, those are all sterilized controls. So basically, the opposite of what we thought would happen happened. So the fracking fluid had the fastest degradation rates and actually degraded to the furthest extent. So if you look at the black squares, that's the synthetic fracking fluid. It degrades pretty fast within about 3 weeks. The revert flow and the polypropylene glycol treatments showed lower overall degradation. So maybe these individual chemical additives, if they're concentrated, they might be more hazardous than, say, the frac fluid itself. In my abiotic sterilized controls, we see 12 to 32% volatilization or absorption to the soils. And then I also tracked cellular growth over time. So in C, that is the biotic bottles, and then D is the abiotic sterilized controls. Day 0 are the speckled bars, and solid bars are day 50. So you can see in the abiotic controls, there's very little growth, or the microbes actually died over time. And then in C, we see that these guys are really growing. They're recovering from any initial shock at the beginning of the experiment. And cell biomass increased about 6 to 14-fold throughout 50 days. So if I move on to the detection of acetone in my bottles, what's interesting is that in the propylene glycol monomer, where we might expect that acetone buildup to be more evident, we didn't see acetone buildup at all. So it suggests that at least the propylene glycol compound that I was using is not going to contribute to the acetone production. But we did see, as propylene glycol is being degraded, we see the corresponding increase in those other metabolites that we were tracking. So propanol, propionate, those compounds were increasing. Again, we saw that acetone buildup in the fracking fluid, which is what we would expect, and in both the corrosion inhibitor and the revert flow. And some data that I'm really not going to get into today, but that I've also looked into is the degradation of specific surfactants, so C8 and C10 ethoxylates, how those are degrading over time. And those degrade actually very fast, within about 2 weeks as well, under anaerobic conditions. So, to summarize, some inhibitory factors: high salt, higher concentrations of dissolved organic carbon typically slow things down, lack of oxygen, systems with low microbial biomass. But what is promising is that microbial communities are very capable of degrading these compounds in synthetic fracking, in our synthetic fluids. There's, I mean, between wastewater sludge, groundwater, they're very capable of degrading organic compounds. Under surface and shallow subsurface conditions, the rates of degradation are pretty fast, within about 35 days at the maximum. In Dan's studies, it was a lot faster. And we see that individual chemicals may degrade slower than the complex fluid itself. So the additives, revert flow, corrosion inhibitor, they degrade slower, which is interesting. So right now we have other incubation studies underway, looking at things in the deeper subsurface, as well as looking at microorganisms present in shale. And with that, I will take any questions. Yeah. Okay.
Any questions for Katie, please?
I got
Yeah.
it. Okay.
Rudy?
Have you worked with any of the frac chemical companies? Have you presented this to some of the suppliers, looking at alternatives or I mean, it sounds good to me what
Yeah.
you're finding.
Yeah.
From our standpoint.
Right. I have not. But I'm, oh yeah, sorry. I have not presented to the chemical manufacturers, but we do have our chemicals supplied. So yeah.
I have a question. And how does research like this, highly technical research, find itself into a greater public awareness or into the regulatory community, so that they have a better understanding of being able to distill between or distinguish between? The question that I had was how does research like this find its way into a broader public awareness or into a broader regulatory awareness, from the standpoint of allowing or having greater educated decision making from a policy standpoint? You know, do you see that research like this, you know, eventually finds its way into, you know, appropriate officials at the Environmental Protection Agency and in regulatory agencies, so that they have a better understanding and can distinguish between fact and fiction related to these types of things?
It's a good question. So the work from Shuai and Dan and myself has been going on for a few years, and really the main way that we can show this work is through publications. So for Dan's work, it just was recently published in Biodegradation. It's just a journal. I think that that's the main way. So Shuai also did a lot of biodegradation work. And then, of course, like, my stuff is not published yet, so it's kind of in the works. But Dan's is the main paper that's come out with our synthetic fracturing fluid recipe. That's probably the main way. And then, you know, we give talks every now and again at conferences. So, yeah.
Mike.
You said earlier in your presentation that you couldn't get frac fluid samples. Who did you talk to? How did you try? Why did you have to develop a synthetic fluid?
Well, I think one reason for that is, for us, fluid samples were limited, and the other side of things is we want to make sure that our experiments are consistent between students, between you know, my experiments versus Dan's experiments. So if fluids that we get from, say, the field are a lot different in those elemental compositions, or they use different ingredients. It might affect our results in the end. So it's just a repeatability, consistency approach. But
you compared your synthetic versus...
Yeah, so initially we wanted to make sure that our fluids were comparable, at least in our additives and our total organic concentrations. So we compared those at the beginning, and I showed one table, but it was more extensive than that. Measured many, many different things. That was a lot of what Schwei did as well.
Dave?
Yeah, let me first of all compliment you on a very nice presentation.
Thank you.
Secondly, would this lead to possibly developing additives to frac fluids that would enhance the biodegradation potential? Things that would sort of accelerate that, and that would make us possibly more environmentally friendly in terms of if there was a spill or contamination, things we could add to the fluid to make it even better.
Right. Yeah, I could see that happening, especially if a lot of these spills are very small. They're not huge releases, but in the case of something like that happening, you could add, so like acetate, for example, that we use as a positive control, that's been added as amendments in contaminated aquifers, for example, to spark microbial growth, and it can actually enhance the biodegradation of other more recalcitrant compounds. But it's this constant balance of course, not wanting to use a ton of hazardous chemicals in the fluids, but also we don't want to use a ton of super biodegradable chemicals that might lead to microbial fouling of our steel or other materials down the well.
Any other questions? Thank you. Good, ready? Thank you, Katie. Come on up. Our next presenters fall under the category of certainly need no introduction. Our friends Kim Collins and Marvin Combs from the Division of Oil and Gas in Frankfurt. Those of you who have followed the activity and the advocacy of KOGA in Frankfurt over the last 6 months, 12 months, certainly know about the passage of Senate Bill 186 in the 2015 General Assembly. Very significant piece of legislation that put into place a significant set of reforms of the regulatory framework for the oil and gas industry. The statute bills passed went into effect June 24th. Following on the passage of the legislation was a regulatory promulgation process to further define some of the parameters and specific requirements of the legislation. As a matter of fact, that regulatory package had its first hearing in committee yesterday in Frankfurt. It will have a second committee hearing in August. Do you know the date?
It's August 6th.
Yeah, August 6th. And certainly don't expect there to be any significant issues with our second committee yesterday. The committee heard the regulatory package and passed it without any questions. And so then is it 7 days after that, Kim, that it officially goes into?
Upon adjournment
okay, so
of that second committee.
So with the fact that there have been some pretty important changes, certainly applaud Kim and Marvin for having done 3 field meetings. Was that June?
June 2nd.
Yeah.
It was,
In Barren River, June 3rd, in Western Kentucky, and Henderson, and June 8th and June 11th.
Yeah, so Barren River, Henderson, Prestonsburg, all in about a 7-day period. to raise awareness and be a resource for what these changes